So we're going to talk now about the equipment of translation, how the cell actually accomplishes translation. So the first thing we need is an mRNA, of course, and this is coming from transcription. So again, this was made during transcription. We also need ribosomes. So ribosomes are going to bind to the mRNA. And then we also need TRNAs. Every amino acid that's included in the protein is going to need a tRNA to bring in that amino acid. So for example, if there's 100 amino acids in my protein, then I'll need 100 tRNAs. If there's 200 amino acids in my protein, then I need 200 tRNAs. So every tRNA is going to come with one amino acid. There are 20 different types of tRNAs. So why are there 20 different types? Well, because there's 20 different amino acids. So each tRNA is going to bind a different amino acid. So we'll start by talking about tRNAs. So tRNAs come from genes, just not structural genes. So they're made from the DNA. So in a process where DNA is read to make tRNAs. They're made out of nucleotides, so the whole thing is A's, U's, G's, and C's, and each one has a different sequence. So for example, tRNA serine is going to carry serine and has a specific nucleic acid sequence that it uses for that. Their structure is like a little L, and you'll see this in some of the animations that you're going to look at. And at one end of the L, you have the anticodon. So notice down here, that's the GGC in this case. This whole thing is made out of nucleotides. We're just highlighting the anticodon. And over here is the attachment site for the amino acid. So one side of the L has the anticodon, the other side of the L attaches the amino acid. tRNAs are going to cycle between being charged and not being charged. So what that means is like loaded or unloaded with an amino acid. So I have my little L, and so sometimes it's like this without the amino acid, and sometimes it gets loaded or charged, and that means the amino acid is added onto it. So this is a charged tRNA. This is an uncharged tRNA. So again, notice that the difference is it does not have the amino acid on it. So that means that this is a chemical reaction that has to be carried out by a whole set of other enzymes. So for every tRNA, has a different enzyme, and remember we're talking about the 20 different types, has a different enzyme that attaches the amino acid. So some people call this the second genetic code. All that means is that this is an extra step that also has to go right in order for us to make the right protein. So there has to be a perfect match between this tRNA and serine. A different tRNA should be matched to methionine, for example. So if this tRNA is going to become charged and have methionine on it, then remember it must have a UAC anticodon. And again, that's to match the AUG codon, right? So some tRNAs have UAC, other tRNAs, like this one we were looking at, has GGC. So this one has GGC in this position. So it's not going to code for methionine. It's not going to match methionine. So again, it's called the second genetic code because there has to be a perfect match between the tRNA and the amino acid in order to get the right sequence. Ribosomes are our other piece of equipment. So the ribosomes are actually made in two parts. There's a small subunit and a large subunit, and they actually float around separately. So here's my small subunit floating around. Here's my large subunit floating around. Here's another small subunit, a large subunit, and then here's my mRNAs floating around. What's going to happen is that the small subunit, the mRNA, and the large subunit will get together. and this will form the functional ribosome. So everything starts out separate. The small subunit and the large subunit will come together with the mRNA to make the functional ribosome. When that happens, so here's the small subunit, so small subunit, large subunit, mRNA, it actually creates 3 different sites called the A site, the P site, and the E site. So 3 distinct locations within the ribosome. These locations are going to be important when we look at how translation happens, because tRNAs are going to travel through the ribosome from the A site. So they start in the A site, they go to the P site, and then they go to the E site. So tRNAs, again, they travel first into the A site, then to the P site, and then to the E site. Once they get to the E site, they leave. So E stands for exit. Okay. So again, it first goes into the A site, then the P site, then the E site. Some people remember this as APE. So just like transcription, there's 3 stages to translation: initiation, elongation, and termination. Initiation is when the mRNA, the tRNA, and the ribosomal subunits assemble. Elongation is when we actually add amino acids and make our long protein. And then termination is when everything stops after the ribosome reaches the stop codon. So we'll take a look at these here. So this is initiation. So here's the mRNA. The first tRNA, which remember should be matching AUG, is also binding to the mRNA, and then the large subunit is going to sit on top of that. Once that initiation occurs, the elongation will happen, and during elongation, new tRNAs will come into the A site. So notice this is where a new tRNA is coming in with an amino acid. This amino acid will get added onto this long chain, and then this tRNA will move to the P site, and this one will move to the E site. So this one will move to the E site, this one will move to the P site. And then this one will leave empty because they're going to come in charged and leave uncharged. So notice that this one is leaving uncharged. This one is coming in charged. Once the ribosome is sliding along here, it's eventually going to reach the stop codon. The stop codon, once it enters the A site, a release factor binds to it. So the release factor binds to the stop codon, and then the complex falls apart. So everything separates. So again, we see that happening here. The protein is leaving, the mRNA is leaving, the small and the large subunit are separating. So the steps of translation sound really complicated on paper and when you try and describe them. But when you watch an animation, they're really simple. And this is a really good animation. It's posted in D2L, and you absolutely must watch it in order to understand translation. So I'm not going to talk any more about this because you're going to understand it so well once you watch this animation, and I suggest you watch it a couple of times. So that wraps up translation. Make sure you're really practicing how to do these problems where you get a protein sequence from an mRNA sequence and a DNA sequence, and we're going to do that in the lab as well. And make sure you watch lots of animations.